I'm
I
trying.
am.
I recognize people's names and
I'm gonna call
people.
it
It's
another
the
10
fish
meters.
and the corals.
020.
Yep.
Roger.
That's going to be a challenge for me. So Tom, what
Well,
we're
just fake
doing
it.
is we're going right. That's right. We're following this contour line between waypoints A and J, AJ and AK. That's John and Kappa. And we have about another 800 meters to go to get to waypoint AK. So
Yeah, and
we
that's
can follow this.
that's crow's flight. So we'll it'll be a little bit more
Yeah.
as we follow this contour.
Yeah, and
Sounds
I
like a
think
good
we
plan.
have. Yeah, I think we have time
Yeah.
to make it to that waypoint and then go up to AL and then make it to the surface. Correct? By the 8 p.m. watch change.
Yeah, I think we should be able to do that. Let me just calculate the distance.
Right, and if we need to shorten a part or maybe not make it to AL, I'm sure that's fine.
Yeah, great.
Yeah, that'll be totally fine to cut short. But what's our time from 200 meters to the surface or on deck?
Yeah, so we usually assign at about 15 meters a minute. So it's a puddle jump almost up to the surface.
Okay.
Yeah.
So we'd have to pull about 7:30, 7:30, right?
Yeah, we
Is that my
usually
calculation
factor
right?
in about 15 minutes for kind of
Extra.
deck work and things like this to get the vehicles on deck. But
Okay.
reasonably, we could probably get it up within 30 minutes, maybe 45 to be
All right.
conservative.
Yeah, so we'll plan right now on 7:15, and then we'll see what happens.
Roger.
Okay, I'm going to do another 020.
You can make them 20 meters if you like. Yeah, and science, let me know if you want any zooms on things as we go by them. We're pretty shallow, so we're pretty responsive.
Yeah. So Abigail asked if we could zoom on one of these white plate-like sponges.
Sure. To the
I think
lower
this
right.
one sort of, yeah, to the lower right.
Roger that.
Thanks, Abby, for the question.
And Abby, if there is something you want to have sampled, let us know.
So Abby, do we know what species this is of sponge?
All right, Marley, you want to go ahead and tie it on it? Roger.
Ooh,
Oops, sorry.
not just a plate.
I can sit down if you want me to be a little less bouncy.
So Abby says it looks like a carnivorous sponge to the right. I think that's the more beige orange sponge that seems to have a candy cane colored shrimp on it.
Marley, you want to come a little wide and I'll sit down a bit. That's great there.
Hello, this is Abby. Hopefully I'm not booting anyone off
wide
by
there, Marley.
signing.
Oh, no. No, that's fine, Abby. It's just Tom and myself.
Okay, fab. So yeah, that looks like
Chad.
there's a...
Go ahead, Marley.
I do not know what that... that looks like two sponges, one with a sort of veiny pattern, those channels growing on top of, actually possibly even three types of sponges that are growing together, and then a little coral underneath. There's also a little carnivorous sponge to the right, I think, which is definitely on our collection list, and we didn't collect any during Express at this
Do
location.
5 meters back.
We know.
Or I can-
Oh, okay.
Alright.
So you'd like for us to collect that
Yes,
carnivorous
please.
sponge?
If we're in a good spot to collect, yes, please.
Sure thing. Are you gonna come lie there, Marley?
Cool.
Make sure I get myself set up.
And possibly just zoom on it, because I'm not totally sure. I just saw it, and I think
Oh
it's
yeah.
that one to the right of the screen now.
The soft
Oh, the stocked.
white guy
Yeah, the little white guy.
above the squat lobster.
It's behind the squat lobster.
Yeah.
It looks like a white stick.
Oh, the one to the
Yep.
bottom right. Roger. That one there.
On the lasers.
Yes.
Marley, you want to do a
Yep, on the
push in?
lasers.
Awesome.
Thanks, guys. You can actually back it up.
Yeah, I will.
A few meters.
And so what is
All
the
right, Marley, go ahead.
species name, Abigail?
So I don't know for sure the species. So you can see, we know that it's a carnivorous sponge, kind of in the family Cladorhizidae, and there are a number of species of Aspestopluma and Cladorhiza. Most of the Aspestopluma are unbranched like this one, but there are 8 species, I think, that have branching morphologies. They have a particular spicule type that tells you kind of what genus, and then allows you to put it into the correct species as well. But you really need to look at those under a microscope to identify them.
So would you like us to set up for a sample collection, or
Yes, please.
roger that? Go ahead and come wide, Marley. Thanks, Marley.
These carnivorous sponges were first discovered in Mediterranean cave. I think it was in the 90s, and since then we've discovered, I think there's over 100 different species.
Wow.
They don't filter to feed like most sponges, pretty unusual. They're often found in nutrient poor environments, so I think that it's an adaptation.
And this would probably be a good candidate for a slurp. Otherwise, it might be a bit small. If that's all good with you there, Science.
Whichever you think is.
Sure. Marley, you want to get a tight zoom on this guy before we sample him?
Sure thing.
What I'm thinking is that we grab the base and then suction, and then I'll bring him to the suction, and then we can slurp him up from the suction hose there on the porch. Oh, sorry.
Yeah, if you can.
Went down.
Sorry.
No, you're good. There you go.
Perfect.
In
Yes,
this,
just
in
right.
this light and angle, it practically disappears.
So what I'm thinking is that we can snip it at the base, and then I'll hold it in front of the suction tube, and then I'll hopefully it'll go so elegantly. All right, Marley, go ahead and come wide. And from my understanding, we have something in the flush.
Yes.
All right. Is that a sample that we want to keep, Science?
It looks like it's just a piece of a sample from 46.
Okay.
So I'm not sure
Yeah.
what the leads want to do.
I would
I
say
think
you
we
could
got-
dump it because I think we have it.
Yep.
Or do you think we should keep it, Abby?
No, I think that's right, Tom. We had a piece of it, and that was just one that didn't make it, so we can get rid of it.
So,
Roger.
and then Meredith asked, before we start sampling, so Meredith asked if we could get a Niskin the next time we are sampling. Is this, if Meredith is still on, is this a time that you would want us to do that?
Well, we can continue
Thank
to
you.
sample and then we'll take the Niskin
Okay.
and she does want it, so.
Yep.
Sure
Okay.
thing.
And Mary, is this going to be sample 053?
Yes.
Okay, great. Me? Yep.
Hey, Marley, you want to push in there, please?
Sure thing.
That's great there. And Kim, would you mind putting on the porch lights, please? Thank you.
So for all you watching, if you look at the little feathery thing to the right, those are hydroids.
All right, let's take it all. Oh, well, hello. There we go.
Nice.
All right, Marley, you want to come a little wide? That's great there. And sorry, Mary, so what is the sample jar that we're aiming for?
Sample jar 3.
Sample jar 3, Roger. I'll get us lined up here. Actually, let's go this way. Kim, would you be able to zoom in there a little on the bubble? Thank you. This angle is not terribly good, is it?
Close.
Roger. Marley, you want to come a little wide? Thank you. All right, Marley, go ahead and push in there, please.
Let me know when you want to start that.
Roger. All right, go ahead and start it. We'll see if it takes. Looks like it'll go. Stand by.
I don't know. It's hard to tell in the brow if you're in or behind.
I think I'm in. Oh,
Nope.
I was.
No, you're out.
Now I'm out.
Right there.
All right, let's try that again. All right,
Like threading a needle.
there we go.
Gone.
All right, Marley,
Yay.
full wide, please.
Nicely done.
Yeah,
Jar.
very well done.
Good job.
There it is.
There
There
it
it
is.
is.
Nice.
Brilliant.
Thanks, Kim.
Turn off the blender.
Roger that.
So just to the left here, there was also the sponges growing on the dead coral skeleton and a little red fan. So if possible, we would like to take a sample of the red fan coral and also some of the sponge that's above it too.
Sure, I'll set us up for over there.
Thank you.
And then after this, you guys wanted a Niskin as well?
Yes.
Great.
Yes, please.
Roger that.
And if we could get a zoom as well before taking the samples again, that would be fantastic.
Sorry, I didn't catch that. Can you repeat?
Just saying, if we could get a zoom as well on the coral and the sponges before we take a sample, that would be fantastic.
Oh yeah, sure thing. Not terribly steady here. Try that. All right, Ben, you wanna
Stick lock.
stick lock? Yeah, thank you. All right, Marley, you wanna push in there, please? And you wanna see the coral behind it, is that correct?
I think it's right under
Correct.
it in the shadow.
Yeah, won't be able to really move the shadows around unless we move the whole rock. Or the vehicle, I suppose, but this will be your best sampling opportunity here.
Good luck.
Roger. All right, Marley, you want to come wide? All right, and so we're going to go for coral and also some of the sponge associated with it.
Yes, please.
Sure thing.
So the coral, and then there's that kind of the light sponge that's growing in sort of a plate shape that's hanging off what looks like a dead coral skeleton. If we could grab a piece of that.
Okay, sure. I'll start with the coral and then we can sort out the sponge.
Thank you.
Sure. Kim, could I grab the arm and bubble there, please? Thank you. Ooh. Marley, full wide? Thanks. All right. All stable there. A little bit of an awkward grab this. Marley, push in there, please, a bit. That's great, thank you.
This view is not going to be a huge help.
You could try to scrape it off and then slurp it.
It might get caught in our slurp chamber. It's a little big. Let's see here. I can just try poking at it. How about that? All right,
Sounds like a
Marley,
plan.
go ahead and push on in there, please. That is wonderful. Thank you. Right. Change my grip force. Yeah, I'll try scraping it off, he said. to push off.
Sponge is pretty strong.
Sponge is going for a ride, isn't it? Huh. That is a feisty coral.
It looks like it dislodged a little crab, and you're like, "Oh, I'm getting out of here."
Oh, I would too.
Yeah.
All right, well, I'll just grab it here then, I guess. Might not be the...
I think it's thinking that claw is bigger than mine.
Yeah.
I don't even think I got a chunk off that, did I?
I think we might have done the classic thing of pointing to the most awkwardly situated coral to sample.
I want to... This guy's really stuck.
Small but mighty.
Yeah.
Well, we can find a better one if we can't get it. Give it another try, and if not, we'll just take the sponge and move on.
Roger.
So this is a Swiftia pacifica coral?
Or a Swiftia kyphoidei. That's why we're collecting it to try to figure out what it is specifically.
Right. Well, I was mostly asking so that others could follow along, and I'm also trying to learn.
Yeah, so this is a group called
Sorry.
Swiftia, and it has stick forms and it has these fan forms. And both the stick and the fan forms are very hard to tell apart on video. You actually have to look at the sclerites under a microscope to really ID them. So that's why we're trying to collect to see if we can figure out which species this is, and if there is some way that we can then equate that to video at some point to be able to distinguish them.
Oh, good.
Right at the base.
Oh, it moved.
It's twisting or twisted.
Great job. Got it.
Hooray.
Yeah,
Nice.
that is
Nice.
amazing.
Yay.
Well done. Jessica, right?
Yeah, that's... Yep.
Yeah, great job.
Thank you. Marley, you want to push in here on our sample?
Yes, our hard fought sample.
All right, that's great. Go ahead and come forward. And Mary, where are we putting this?
How about starboard D?
Starboard D, Raj. Thanks, Kim. And Mary, before we open, do we have anything floaty?
No, we should be good.
Roger.
That starboard vertical is blowing.
Yeah, blowing in the breeze a bit there. If you want, we can do the same kill technique that we did before.
Yeah, might float away, but
I'll let you know when to kill. Oh, that is the wrong one.
all right, you're really going for a scoot.
Yeah. Ben, you want to press stick lock there?
Yep.
Thank you.
Dismount.
All right, it's in there. Thanks, Kim.
Thanks.
Would you mind closing up the bin while so I don't do some bad things to it? Oh, all right. Hard down. That way, you want to keep coming up there, Kim? I think.
Yeah,
Or
I
thank
still got
you.
the starboard vertical off.
Oh, Roger. Thank you, that'd be why. Thanks, Kim. That makes sense.
Resume normal breathing.
Resume.
So we're going to go back and collect then the sponge that was there.
Yeah, we'll collect the sponge and we'll take a quickness skin. Just need to drop
Okay,
down in the
great.
water column a bit.
So Abby, do you want the light colored disc, the white sponge with the webbing in it, or did you want part of the base?
So I think part of we have a lot of sponges on the list that look like that kind of white plate sponge. We don't know what they are, so I think it would be great to get a sample of that. Preferably.
Kim, you want to keep coming down a bit, please?
Yep.
Sorry, I think I keep getting pops on and off SPL. I was just saying, I think it would be great if we could get a grab of the sponge that looks like a white plate, especially if we could get it where it's not touching either of the other two types of sponges, because we do have quite a few unknown demo sponges with that look similar to that one in our imagery.
Roger that.
Roger.
Thank you.
All right, sorry about that.
The original target is 250 from you. But
250, Roger.
Yep.
All right, we got back to our plate.
Brilliant.
That sponge doesn't have a coral underneath it.
I know.
It lost its buddy.
All right, did you want this sponge to go in the same bin as the coral sample?
That would be, yeah, that would be fine.
Sure thing.
You try and keep the sponges separate, but they can go in with corals, no problem.
Sure thing.
Poor crab down below is all covered in sediment now.
Oh yeah, he's probably having a day. Interesting. All right, Ben. See how we settle out. Okay. And do you want a particular part of the sponge or just anywhere along it?
I think if you could go aim for that top edge, just because that seems to be farthest away from the webby looking sponge that Tom was talking about on the left, and then the sponge growing along the base of that coral.
Okay. Riley, you want to push in, please? Thanks. So somewhere kind of over here in this upper edge
Yeah.
here?
Yep. Yes.
Okay, great. Great, thanks, Marley. Maybe I'll do actually a little bit lighter grip force.
Perfect. You made that look so easy.
All right. Marley, go ahead and come on wide, please. Oh. Unless science wanted a picture of the sample. It's mostly in the jaws, though.
Yep, that looks good. Thank you.
All right, great. Maybe I'll just get my hand deep enough in there and I'll let go. Oh. Looks like a floaty bit. Mary, you want to keep an eye on that floaty bit? I think it's going to land an E. Broke off there from the sample.
Okay.
All right, looks like it's gone. Thanks, Kim.
Thank you. I think that floaty bit might just have wafted out of E.
Yeah. Oh, wafted out E. Roger.
Possibly. But thank you. That's great.
All right. And to confirm, we also want a Niskin here before we kick off.
Yes, we do.
Correct.
Great. Mary, what Niskins we got open?
All of them are open, so how about we go with Niskin 1?
Roger that. And Marley, you feel wide there?
I believe we fired one last one. So I think one is already used.
All right, let me
Yeah,
check. Yeah, it just wasn't
And
marked on
Meredith
the...
steps off.
It
Cool.
It is used, it just wasn't marked on the sheet. Thank you.
Cool. Yep.
So go for
yeah,
two?
we have
Two.
it on.
Sorry, Lisa, what was that?
I was going to say that's what we have on our data recovery report. Shows that Niskin 1 was used on the 12 to 4 shift.
Roger.
True it is.
All right. Two bundles there, am I?
You don't get any easy ones now.
Oh, it's good practice for me, so I'll take it. Hey guys,
After you got the
eyes?
coral, you can do anything.
Pop.
Got it.
Roger. Thanks, guys.
And could I confirm that sample number?
That was sample 056.
That's what I have. Thanks.
Thanks, Kim.
So could one of you explain what just happened? So I think Jessica pulled on a cord and that fired the Niskin bottle. It's sort of just, that's how it's done.
Yep.
Yeah, that's exactly it.
Oh, cool.
We have Niskins
So
rigged on
each of
the...
those,
Oh, sorry.
right. So each of those cords corresponds to a Niskin bottle.
Yes, that's correct.
The color. Ah, that's cool.
All right, so we're going to just continue on this contour and
Yeah, I'm going
flying
to keep
along.
on 020. It might become 015 slowly.
020.
Let's just do it small and steady.
Roger that.
Yep, we'll just head toward the next waypoint.
Great.
Quick over you, Kim.
And let us know when you want us to start a transect if you want certain spacing between the last transect and
Yeah,
this.
exactly. Because we're not, let me see if I can find the target. So,
Yeah, we need to be about 100 meters past the last one.
Roger, yeah, we're like 70 meters.
2? Yeah. Oh,
1 meter.
that's farther than I thought. Yeah, I thought it was 1 or
Yeah.
2, but and for everybody out there, the Niskin is a way to collect water at a discrete depth and location. It's basically a tube that's open at both ends, and when you pull that cord, it releases the two ends and they get pulled back in with like a rubber band so that they're held tight so we can get a water sample from this depth. And we use that, right now we're going to use it for environmental DNA. So you hear us talk about eDNA, which allows us to look at the DNA that's floating in the water and see all the different creatures that we're not seeing on video. And some of them we will, but some are just off camera, and then we can actually pick up what's there by using the eDNA.
Yeah, and I guess with the eDNA, we can figure out what species are in the area that tend to be more cryptic, especially if we have voucher samples for those cryptic species.
Yes, we can do that,
Those species.
and we can also tell, like if the coral that we collected, we may be able to tell what species are there just without seeing them or not being able to visually ID them. But we can then tell what's there. And now we have the fish staring at us.
Yeah, we're just watching out for dinner. So, back up here.
Well, that's the next trick. If you can use the manipulator arm and grab a fish, that's when you win.
Is that shiny object, is that a fish there on the bottom?
Hard to
That's
say.
a great question.
Can have a closer
Yeah,
look. Looks like it was a fish.
Okay, if you grabbed it, then you will have won, according to
I
Tom.
guess I'd have to specify differently then.
A live
It
fish.
does look like the
You're
carcass of a fish. Looks like a dead ratfish because of the way the tail is. Could we take a look at the coral to the left, the white one? The white one on the rock?
Yeah, Roger. A guy there below the lasers?
Yes, that one right there. We could zoom in on that.
Yeah, go ahead, Steve.
So this looks like the Caligorgia that Meredith and I were talking about an hour ago, and we finally get to see it. And it has one of these black coral crabs on it. That's what I call them, because they live on the Christmas tree corals, and they kind of look like spiders. They're different from a lot of the other, or similar but different from the other crabs that are down here on the bottom. That's good, thank you.
Okay.
So Steve Oskovich confirmed that that's a Caligorgia.
Okay, we're heading 015 is our
015, right.
general vector.
I turned around to look at the coral there.
Raj.
It's a rather alarming disparity between the Argus altimeter and the sub-bottom profiler.
Quite alarming. Don't want that.
Those
I wonder if it
tether pings, I think.
is it.
Yeah.
Yeah. It catches it, and then it jumps back to 21, and then 999, and all over the place.
it looks like it's bouncing up and down.
Yeah.
Oh yeah, wow.
I liked it better when you were in 6 meters.
Bridge nav. $20 meters, 015. Thank you.
Wow, top of a cliff here.
Where all the good stuff's going to be.
Yeah. Oh.
Is that a black coral there that we're passing? Or what kind of coral
Yes,
is that?
Right. The two big bushes are
Yeah.
Christmas tree corals. They're black corals. So you can see their skeleton is here. It just looks dark, but it's black if you zoom in on it.
So I'm trying to recall, Tom, isn't that the species that you and Mary wrote a paper on that you discovered for the first?
Yes, Mary and Milton really wrote it. I was involved, but they get to be about 3 meters tall, and they're just super huge, basically. And people they'd caught them in trawls and things, but no one ever described it before. So they were able to describe it, and it just shows that there can be huge things that are at the bottom of the ocean that we just don't even know about. And this is right off the coast of a very populated area.
Right, right. Where a lot of commercial and recreational fishing takes place.
Yes. Could we zoom in on the white of that coral, the coral that's on the coral?
Yeah, go ahead, Steve.
No, that
That looks like Lophelia.
No,
Is that what you were going to say?
no, it's an octocoral. I'm thinking it's an anthothela or anthotheelid.
Oh, maybe, yeah, because it does look softer.
That was one of the ones on the list, but it's kind of a tough collection, right?
Huh. Easy,
Yeah.
easy collection.
Don't challenge Dan like that.
Let me go back and check the
Well,
list.
Christmas trees are tough to get.
We're already landed.
Yeah, anthothelia
You could
species.
scrape some off.
Sure.
Because Christmas trees are
What about
tough to break, so.
Could probably, I don't know if it'll suck. Oops, sorry. Will it suck out of there with the slurp gun?
It's not a fragile one. It's one of those flexy ones. So maybe.
I could try and probably be easy if I try and grab a bit, see what happens.
You can also try scrape and slurp too.
Yeah, I could do. Scrape and slurp. Apparently
Fancy terms.
closer than I thought here. Want to zoom right in there, Steve? And can you turn the porch light on?
Yep.
No, not you. How much do you want? Is a little bit okay?
Yeah, you could probably 10 polyps, so a squeeze in there, whatever you can get.
I don't know what we'll do with it after that, but Maybe I can get the branch that it's on. Something like that.
Yeah, I mean, that's pretty good.
That'll do it.
Merritt, if you need more than that, you can chime in.
There's another branch there. Another dead branch.
She said it was a great grab, so I think she's happy with
Copy.
that
Awesome.
amount.
You want to slurp that, or you want to?
I think we have, is D still open?
E is open, the starboard outer box. And
A, B, C, but D, one of the little boxes is not open?
No, they're all full with corals.
I can try and
Can
feed
you
it to
slurp
the
it?
slurp. Yeah. Okay, let's switch over to slurp view on the cameras there. Steve, you can zoom back in while we're waiting.
I can't tell if that black coral is attached or if it's just along for the ride.
I think it's, well, I kind of grabbed it and snapped it off, so the little guys were on there. You want to go to the flush jar and we'll flush?
Yep.
Slurps have been working okay, right?
Should be. Okay, zoom all the way in there, Steve, see what happens.
Flushing right now.
Yep, go for flush. And then what jar are we going to stick this guy in?
Number 4.
Number 4.
And this is sample 57057.
Okay, that looks pretty
That is correct.
flushed.
Thanks.
Air flush is stealing my flow. Okay, you want to turn it off there and go to jar number 4.
Yes.
Jar number 4. Let's flush there for a second. And Steve, you'll have to zoom out. I'll need the camera to feed the slurp here. You can zoom right out for a second until I get oriented, thanks. This is gonna be backwards. Okay, you can zoom in again. That's good, thanks. Is the slurp on, is it?
Yep, it's on. I can put it up too.
Go full power there. Can you also put, oh, you got it, bubble cam on, look at you.
Yep.
Okay, does that look like it's lined up there?
That looks like it's in.
Yeah, no, yeah, it must be.
Yeah.
Oh,
Up
it
she
went.
goes. All right, back off to 50%. Can frame up the shot again there if you want. What's the yellow stuff? Oops, sorry. The yellow stuff on there.
Have you seen it go through yet, Steve?
I didn't see it yet.
No, negative. Still waiting. Are you at 50, are you?
Yeah, 50.
We'll bump it up just a little bit. Go full power for a second. If that doesn't work,
Roger.
I'll play with the hose.
Full jam.
Steve, can you zoom out? I'll just give the hose a little deal in, out, in, out. I can zoom right out. Oh, thanks. Ah, it is the camera that blocks it. See that shadow?
Yeah.
Mm. Yeah.
Or maybe it's just blocking the porch light. I don't know.
Yeah, I think that's with the porch light.
Still hasn't come through, right?
Nope.
And grip force 9.
There it is.
Yay.
Oh, okay, you can back off, kill it.
Good to know that's working again.
Nice.
Yeah, there's a spot in the hose where it goes from the larger hose to the smaller hose, and stuff likes to get caught there. Okay, you can go back to the flush jar, please. And disable that thing if you would, please.
Yep.
All right, what were we, 3 something? 0 something?
015.
015, thank you.
And how far we moved from the last transect?
Let me just double check that for you. I don't have a mark here. I'm presuming it's this mark, which is unlabeled.
That could be because it happened during watch change, so.
Yeah, about 100 meters since the last.
All right, let's go 20 meters more just to make sure, and then we can start a transect. Same direction.
Copy.
This is the Charlie Brown Christmas tree.
Totally. Can you center up the bubble cam for us?
Yep.
Bridge nav. Do 40 meters, 015. Thank you.
What's this going to be, 100-meter transect?
I think 200 is what they've been doing.
It'll be,
200?
yeah, it'll be about 200. Yes.
Yeah.
So you can go faster if you want while we're transiting to the start point.
Yeah, copy. I'm just getting the playing with the altitude there because it's downhill a little, so we put the